Okay, I'd like to welcome everybody to the August 8th meeting of Glimprint and the IMAG MSM Working Group on Multiscale Modeling and Viral Pandemics. We're very pleased to have Bishal Paudel from University of Virginia talking to us today about cancer phenotypes. As usual, I need to remind you that this meeting is live streamed on YouTube and an edited version of it is recorded and available afterwards for public use. As always, Tom Helikar and and Jim Sluka and Lorenzo are available for your suggestions, comments, complaints. We are always trying to do better for the community. Please reach out to us. We have Twitter, Slack, which doesn't get used very much, the IMAG MSM wiki page, the IMAG LinkedIn page, and our YouTube channel. And there's also the Glimprint website, which Tom and Jim Sluka have been working hard to try to improve. Please help us develop these communities and reach out to people so that these great resources are made available. Again, the Glimprint webpage, Jim has been working on it a lot. Please help us develop it. At the moment, it's still pretty much a stub. It needs love. We have that MSM viral handle also to use as a tag if you're going to repost items from the site. Okay, we do have a few announcements. University of Florida has some open faculty positions in laboratory of systems medicine. That is an incredible team led by Reinhard Labenbacher, who of course was co-founder of this community here. I strongly encourage people to contact him about that. Really great opportunity. There also is Jason Shoemaker has a recently funded grant in computational modeling hormones and influenza infection. That is a topic dear to the heart of many people. And so if you are interested in that, some of that material has been presented at various mini seminars over the past couple of years. So if you want to find out about what he does, you could look at our archive. Please do reach out to him. It's a great opportunity. The IMAG MSM annual meeting has been finalized for the 30th of September to October 2nd. It is going to have the theme of biomedical digital twins. Of course, this community has been instrumental in pushing the idea of medical digital twins. And many of the community members are organizers, and particularly Gary Ann is one of the lead organizers of this meeting. So we encourage you to reach out to Grace and Gary about your interest in participating in this community and that meeting. So do let people know if you want to go, if you want to speak, if you have ideas for breakout sessions and so on, please reach out to Grace and Gary. There is going to be an AI workshop, AI, artificial intelligence and precision medicine in diabetes and chronic diseases, October 30th and 31st. Again, this is potentially of interest to many people. If you want the links, either maybe Jim, you could put some of these links in the chat if you would, or look at the slide deck and you'll be able to find the links. There is also a new well, it's not an RFA, it's a NOSI for systems modeling of infection and immunity across biological scales. Many people in this community have submitted either collaborating or competing proposals for the big U54 National Institute in systems modeling of infection and immunity. This is a follow-on to that for R01 scale and R24 scale and R03 scale grants. I think it's a great opportunity. It is really something that many of us here are interested in. And so I encourage people to look at that. If you have a publication to share that has anything to do with the topics of interest to this community, please do send them to Jim. We could put them up and publicize them on the IMAG site and also the Glimprint site. We're always looking for news and things to reach out and help us keep things up to date. We will have a steering committee meeting tomorrow morning at 11 a.m. If you are a regular steering committee member, you've already gotten an invite. If you didn't get an invite and you think you should have gotten an invite, let us know and we will add you to the list. We're always happy to have people who want to help build the community. The topics of conversation are primarily going to be about trying to organize a in-person meeting to build community for Glimprint, and also a possible workshop on modeling, multi-scale modeling of immune response, a virtual one, more of an educational style one. And so those will be the two main topics for this. We have some possibility of funding, and so we need to think about how to secure that funding. Are there any other community announcements before we go on to our presentation today? Okay, we probably will not meet August 22nd. Usually the dog days of August are times when either the Europeans are on holiday or the Americans are getting busy to get ready for the start of the semester. We will have the rescheduled talk by John Metzgar, translating and applying Boolean network control theory in multi-scale settings, specifically in the context of trying to develop improved therapies, personalized therapies for cancer. We need somebody to speak on September 19th. So if you have suggestions, as always, suggest yourself, suggest your graduate students, suggest your postdocs, suggest the people you would like to hear from. And Lorenzo is extremely good at tracking people down and getting them on the schedule. So please help us for that September 19th meeting and going forward. Dagmar Eber from Zurich, of course, the developer of much beautiful multi-scale models and multi-scale modeling frameworks, will be speaking October 3rd. So I know people will look forward to that. We have a Google form for suggestions, or email them to me, Jim Sluka or Lorenzo or Tom, and we'll try to get our best to get your people on the schedule. Again, if you've spoken before, speak again. If your students have spoken or need to practice a PhD presentation, feel free. This is a meeting where people of all seniorities and a whole variety of topics are welcome to speak. So please do help us fill up the roster for the fall. Okay, I don't do elaborate introductions today. We will have Bishal Paudel from University of Virginia giving us insight into his work on molecular variability in cancer phenotypes. And I think this is going to have a nice balance between methodologies, which will appeal to this particular audience.
Like Jim said, my name is Bishal Paudel. I'm currently at University of Virginia. Before coming to UVA, I trained at Vanderbilt in Nashville, Tennessee, in cancer systems biology group. So my work is going to be focused on understanding the molecular variability that drives divergent cancer phenotypes. So I want to start, and maybe this introduction may not be necessary for this group. When we think of biological systems, we have found that biological systems span several scales, from cells to tissues to organisms. The information flows back and forth in either direction. At each scale, we with the technology that we have at our disposal now, we know the biological systems are highly heterogeneous. So if you want to look at a fine enough resolution, you'll always find differences. What is fascinating oftentimes are the interactions among molecules or even between cells within a multicellular context. Those interactions capable of giving rise to distinct qualitative phenotype that is distinct from the continuous phenotype that are often characterized. So when you think of the noise, this is often described with the phenotypic traits that are normal distribution, but then the interactions that are capable of giving rise to those distinct phenotype, give rise to multimodal distribution. If there is only two phenotype that is distinct, then you see two phenotype, two distribution, and so on. Today's talk, I'll focus on kind of transversing this biological scale and talking about heterogeneity at molecular level as well as at the organismal level. In the first part, I'll talk about some of the work that I have done with Kevin James in UVA, where I'll go from cells and looking at the cellular systems and identifying differences that drive multicellular phenotype. And in the second part, I'll talk about the work that I have done with UVA Cancer Center in identifying the biomolecular measurements that drive differences in clinical outcomes in patient settings. And feel free to interrupt me for any questions as you see fit. So the first part, I'll talk about the drivers and mechanisms driving tumorigenesis. So when you think of premalignant state, it is often described as the controlled proliferation that is constrained within some biological constraints, but some of those premalignancies go on to become malignant, but not all of them. So the question that is driving the cancer field is what is unique about some of those premalignancies that go on to become malignant. So the first part, we'll kind of talk about that in some detail. So when we think of the what gives rise to heterogeneity in from a systems bio perspective, it often comes as similar cells within a uniform environment that could give rise to distinct outcomes. Several synthetic or engineered biological systems have been identified that have been shown to give rise to multi-stable subpopulation in both bacterial and eukaryotic cells. But some of these phenotypes are often described at the single cell level. And in a long-standing observation, which is now about 20 years old, 23 years old, a unique multicellular phenotype that was driving this distinct behavior was described in a very engineered synthetic system. So in this schematic here that I'm pointing to, synthetically engineered HER2 receptor, EGFR receptors have been engineered, which would be activated when you bring the synthetic ligand in, which lacks the extracellular domain. So when the ligand brings in those two receptors as heterodimers, the Muthuswamy et al. Manuscript showed that it gives rise to this hyperproliferative multicellular outgrowth. What is unique about this in the context is this hyperproliferative outgrowth kind of mimics what is the ductal carcinoma in situ, often described in the context of breast premalignancy, which is driven by HER2 receptors or EGFR receptors. When we looked at this in a more systematic way, what we found is not all of these three, and this is done in 3D culture setting. So when we culture them in 3D setting for these synthetically engineered cells in breast epithelial system, we found that not all of them go on to exhibit the similar looking phenotype. So many of them have a roundish, regular hyperproliferative state, but then some of them go on to exhibit this more irregular phenotype that is distinct from the others. And when we quantify them, we see that only 1/3, about 33 to 35% of these 3D multicellular outgrowth go on to exhibit this irregular phenotype. And this irregular phenotype from a breast premalignancy context mimics what would be an early escape from ductal carcinoma in situ to kind of become invasive in the early transformation of breast epithelium. So what is driving only 1/3 of these 3D multicellular outgrowth into this escape from the constraints that they were in has been of interest for many years for both systems biologists, but also for cancer biologists in general. So we found that this distinct multicellular outgrowth that mimic this escape from the DCIS state becomes distinguishable phenotypically at about 48 hours of the receptor activation. So we performed transcriptional profiling of the 3D clones and quantified and matched the abundances of the genes to the phenotype frequency to identify the core set of signatures or genes or transcriptional profiles that were associated with this phenotype. And we found was one of the key biological processes in cells, nucleocytoplasmic transport or protein localization, as one of the enriched pathways that were distinct from that is distinct in these multicellular growth. So from the nucleocytoplasmic transport, when we looked at the regulators of nucleocytoplasmic transport, we validated some of these nucleocytoplasmic transport and their role in the emergence of this multicellular outgrowth experimentally, and showed that loss of function or gain of function of those regulators to actually perturb those phenotype. For the sake of time, I'm not displaying those results. But what we see is among those 3D culture clones, there is significant heterogeneity in the abundances of the initial conditions or initial abundances of these regulators that are involved in the nucleocytoplasmic localization or transport in cells. So to systematically understand how nucleocytoplasmic transport is regulating this multicellular outgrowth or an escape from the premalignancy in breast epithelial context, we thought it would be infeasible for us to kind of perturb each gene at a time. So we wanted to look at more of a systems level view on this. So we reconstructed a mathematical model of nucleocytoplasmic transport from the early works of Riddick and Macara. And this is a ODE-based model that has been published for many years now, and utilizes the key principle of how the cargoes need to be imported into the cells and how they could be exported out of the nucleus into the cytoplasmic region. So one of the key process that is involved is the binding of the cargoes with the importance that are required to bring cargo from cytoplasmic region to the nuclear region. So importin alpha or importin beta, and different cargoes have different binding affinities or different binding preferences in terms of which mechanism of import they undergo. And this bidirectional flow is often mediated by Rayne gradient as shown here. So when we calibrated this model in the context of our experimental cell lines, and we wanted to examine what does the nucleocytoplasmic transport state looks like under these initial conditions, under these initial conditions of different 3D organoid transcriptional states that we profiled. And what came quite interesting was we found this divergent accumulation of nucleocytoplasmic transport. So we are looking at the nucleocytoplasmic ratio or the steady state ratio of the cargoes that have certain properties of biochemical import or biochemical export. And we found under the same initial conditions, some 3D clones exhibit a higher abundances of those steady state localization of the cargoes. We're looking at the classical NLS bearing cargoes, meaning they require the binding of importin alpha and importin beta. And there is this divergent accumulation. So suggesting that maybe there is a divergent nucleocytoplasmic transport reconfiguration that leads to some cargoes that are mislocalized, and that divergence maybe could be contributing to the differences in the phenotype that we saw. When we looked at the association between those steady state localization of cargoes to an exportin, CSC1L, or importin, KPNA1, and a whole set of other regulators, we found that the ratio of those nucleocytoplasmic steady state levels were anti-correlated with the exportin, and then positively correlated with the importance. And this is important because experimentally we found that perturbing the CSC1L, which is also known as XPO2, is a nuclear export, so exports molecules out of the nucleus into the cytoplasmic region, is one of the genes that we found to be significantly altering the phenotype frequency in synergistic way to the dimerizer that we had experimentally used. So this begs the question that we see the reconfiguration of nucleocytoplasmic transport. There is divergent cargo accumulation, but what are the cargoes that are actually being imported or mislocalized in this process of receptor activation? So we did a lot of biochemical assay and high throughput mass spec experiments, which I'm not going to show in detail, but we found that one of those cargoes could actually be the receptors themselves, because when we looked at proteomics, EGFR family of receptors actually came and as top hits, and also the scaffolding protein that are involved in the endocytosis process. So we wanted to see if the receptors are internalized, and if we block the internalization process, which is often mediated by clathrin-mediated clathrin heavy chain or clathrin-mediated endocytosis. So we wanted to examine what happens to the phenotype that we see at this multicellular level if we block this internalization. So we knocked down experimentally clathrin heavy chain and use cell and molecular biology techniques, and examine what happens under different knockdown conditions. So I'm showing two knockdowns where in both of these knockdowns, when you block the receptor internalization by blocking this clathrin heavy chain, we see an increase in the phenotype frequency that we quantified and I described earlier, suggesting that when receptors are somehow prevented from going into the cell, into the nucleus, or into the cytoplasmic region, the phenotype is promoted. So under a normal, under these conditions, what we thought was when the receptors are retained in the cytoplasmic region, in the cell surface level, they promote the outgrowth. But if they are somehow able to mislocalize to the nuclear region, then they repress the phenotype. So which we followed up and examined how they are able to do that. So the other question that we had was what happens and how are the receptors getting to the nucleus first, and also what are they doing in the nucleus? So we performed chip sequencing, which looks at the enriched genomic regions when the receptors are internalized, and we identified some genomic regions of interest. And one of the regions that we identified was a epigenetic microRNAs, which is elevated or increased upon the receptor dimerization context. And I guess for our sake, the epigenetic regulator or the microRNA 205 represses or negatively regulates the expression of importance. So what we settled on was under a normal architecture of this nucleocytoplasmic transport, we have the receptors binding with important alpha or important alpha or important beta, and we saw this through biochemical assays that it requires the binding of important alpha and important beta both, but we didn't know what the binding affinities of these receptors as a dimer is. So we wanted to mathematically simulate this and examine what happens under the experimental initial conditions of both importance and exportance to the ratio of this steady-state localization of cargos, and how that would be related to the phenotype that we're looking at. So under this normal network architecture where receptors get imported into the nucleus, we didn't know the binding affinities, so we changed the binding affinities by range of values, and we looked at the cytoplasmic to nuclear ratio. And when we looked at the abundances of important alpha, there is slight variation in the cytoplasmic to nuclear ratio. But when we looked at CSC1L, which is one of the regulators that we experimentally found to be important in the context of these phenotype, we did not see much effect in the cytoplasmic to nuclear ratio of the steady-state cargos. When we incorporated the newfound information about how these nucleocytoplasmic transport was deregulated, how receptors were internalized to the nucleus to activate the microRNAs, and how those microRNAs were repressing or negatively inhibiting the abundances of importance, with the new architecture that we encoded in the model, we found that the abundances of importance does not really have much effect to the cytoplasmic to nuclear ratio. However, when we looked at the abundances of CSC1L, now it almost acts like a switch. It's very sensitive to the abundance of CSC1L now, where the cytoplasmic to nuclear ratio is very low at the lower abundance of CSC1L. But when it is elevated, then it kind of goes on to be a higher nucleocytoplasmic steady-state concentration of some cargos. So this suggests that this receptor activation creates this cross-inhibitory feedback, which gives like this switch-like behavior in the localization of cargos, and that switch-like behavior could be regulating some of the phenotype that we see. So we wanted to examine whether CSC1L abundances actually have something to do with the phenotype that we saw. So these were experimentally determined CSC1L abundances from immunofluorescence assays, where we looked at the variation in CSC1L at single cell level or the single clone level under both activation of the receptors as well as no activation of the receptor. And we found that there is significant difference in the abundances of CSC1L. But most importantly, we see this when we model the abundances of CSC1L under the dimerizer context or plus AP context into the model with the new architecture. We found that there is a cytoplasmic to nuclear ratio with bimodal distribution. So almost 1/3 of these, the nucleocytoplasmic ratios exhibit a different distribution compared to the other, which we think might be the driving factor for this multicellular outgrowth. So initial abundances of CSC1L, depending on what cells have, their abundances set in the initial conditions might be the factor that may push them into one or the other in terms of committing to this multicellular phenotype that we described. So I thought this was super cool because this now lets you examine how nucleocytoplasmic transport is involved in different cancer contexts. But one of the things that I got immediately drawn to was, can we use this nucleocytoplasmic transport model that we have developed into more like a personalized cancer context? So I saw Dr. Clayton Yates, a racial disparity researchers from Johns Hopkins. His work has shown that depending on the localization of some cargoes, there may be some importance of those localization in the identifying the high-risk group in African-American cohort compared to the Caucasian group. So, but some of these work that he had published, they were very cell molecular heavy and was not distinguishing which way these cargoes would behave. So I reached out to him, and we were able to get some of the patient-specific information from the cohort that he had data on. So I looked at for both African-American cohort and European American cohort gene expression differences in the nucleocytoplasmic transport regulators, and I simulated what would be the transport state like depending on those gene expression states. So this is a patient-specific model that predicts what those steady-state localization of cargos and different cargo types would look like in each patient. And immediately we see that there is different abundances among patients, some enriched in African-American, some in a Caucasian group. But most importantly, what we found is some of the classical NLS-bearing cargos, if they are cargo 1 or cargo 2, which basically have the distinction of if the cargo requires the binding of only important alpha, or important alpha and beta, or binding of only important beta. When I looked at those distinct categories of cargos, I found that for both of these cargo type, there is a higher nucleocytoplasmic localization in European cohort compared to the African-American cohort. Then I wanted to see if we could use this information to identify and stratify patients based on their risk group. So when you think of these cargo signatures that we could extract from this nucleocytoplasmic transport model that is specific for each patient, I simply divided them into a median high and median low. And when I looked at the survival probability for the patient, this is a breast cancer African-American or Caucasian women, I found that the African-American cohort, if they have a higher signature for nucleocytoplasmic localization of some cargos, we are able to identify this high-risk group that are not clearly separated in the Caucasian group. So this work in terms of patient personalized signature could actually have application beyond the divergent phenotype that I described earlier at the cellular level.
Michelle, I have a question. This is Tom. So is this system influenced by estrogenic exposures?
No.
No, okay.
I think the model at its present form actually could be applied to any disease context, and I think that's where I'm kind of thinking of applying this in other disease contexts where the nucleocytoplasmic transport has actually been implicated to be important. And you'll see maybe in the next portion when I'll talk about the leukemia disease, where FDA recently approved for multiple myeloma nuclear export inhibitor, which was very effective for one type of hematological malignancies, but not so effective for other diseases. I think this modeling framework could actually be useful in identifying some signatures that could indicate which patients would respond to some of those transport inhibitors and which patient would not. Because when you think of the nucleocytoplasmic transport, is a very essential biological process, so you don't want to block that process in every cell. So it is useful in identifying and stratifying those patient cohort if we know what the signature looks like. So it has a lot of application beyond the breast cancer context that I described in the first part. Okay, so I'll move on to the next part. So in the second part, I'll kind of talk about different clinical outcomes in more like a inter-patient differences, and kind of come back and try to integrate multi-omic, multimodal measurement, bioinformatics analysis to identify what are the molecular differences that are driving those distinct outcomes in those patient data. So this work is this work was done as part of the research activity for program project grant, PO1 grant to UVA Cancer Center, which is focused on understanding the role of sphingolipid metabolism in acute myeloid leukemia. I'm not going to go and talk about a lot about sphingolipids, but when you think of sphingolipids, they are essential lipids, but they have been shown to have distinct intracellular functions, including cell proliferation, death, and so on. So AML has been described to be very highly heterogeneous disease. Several genomic classifications have been proposed, but this work and this program project grant primarily focuses on looking at the biochemistry of the AML and not so much on the genomic classification. And our objective is to see if we could add biochemical information on top of the existing genomic information to further stratify patient and hopefully identify new therapeutic opportunities. So from a metabolism perspective, the sphingolipid pathway kind of has this very simple schematic where ceramide is the key hub that gets converted depending on the enzyme activities into different sphingolipid species. And really fascinating thing about this pathway is depending on these species, which species are abundant and not, either some of them have pro or anti-apoptotic function. So there is this balance that is critical for cells to achieve in terms of how they regulate cellular function. So this pathway and its balance is very, very important. So in this research activity, what we looked at was we had patient samples collected from 3 different research institutions. We had data from cell lines, and we had multi-omics information collected on all of these patients and cell lines. But one of the key things that we looked at was when we looked at the lipid or metabolite abundances in this patient, often characterized into this sphingolipid group with different carbon chain lengths and so forth. We wanted to examine if we could identify some patient groups that are similar or dissimilar based on those lipid abundances. So we applied consensus clusters and identified some robust statistical clusters that uniquely define some patient and cell line cohort. So for some of these patient and cell lines, we had the RNA sequencing data as well. So we integrated the lipid information with the RNA-seq data to identify some gene signatures, and you'll see throughout the remaining half of the talk, to identify some unique signatures that would be contributing to different lipid clusters that we identified. But the motivation for us beyond this was, can we use those gene signatures and develop some machine learning classifier to be able to apply those signatures in the new cohort of patients that are publicly available and very large cohort compared to the cohort that we have, or any future cohort that we get the data from, in a way that we could connect and associate lipid profile with the clinical outcome. So I developed the classifier based on some gene expression differences that I had identified based on those lipid signatures, and we were able to apply those signatures into two very large cohort of AML in TCGA and Beat AML. So when we looked at the lipid profiles focused on the sphingolipid pathway, we found two distinct clusters of cell lines and patients, and they nicely integrated both cell lines and patient into both of these clusters. We found that one cohort of patient had a higher abundance of this sphingolipid species called sphingomyelin, and the other cohort had a reciprocal abundance of hexosylceramide. So there is this nice difference in terms of the abundances of hexosylceramide and sphingomyelin. So some of the, like I mentioned earlier, some of these patients and cell lines have information on both lipid quantification as well as gene expression. So I wanted to examine whether we could only look at the gene expression and see if those patients and cell lines separate into unique groups. So when I applied global gene expression-based clustering, I found 4 unique clusters, and that is different from what we identified based on the lipid-based clusters. But most importantly, what we found interesting was when we looked at the RNA expression-based cluster, they were either clusters that is dominated by patient's sample or the cell lines. And there was not much of integration of those patient and cell lines based on the global expression of the genes. However, we found in our lipid clusters that there is nice mix of cell lines and patient information. So we thought sphingolipidome, which is more biochemical measurement of the state of the cell, integrates that cell line and patient information much better than what global gene expression would do. So after we identified these clusters, because some of the patient cohort that we had collected, we had clinical annotation available as well. So we wanted to see if those patients that fell into one or the other cohort have unique clinical outcomes or clinical differences. So in this plot, I'm looking at the primary response. So they're often given the standard of care, which is a standard combination chemotherapy. We found that a patient that fall into the sphingomyelin high group and hexosylceramide low group often have a higher rate of failure compared to patients that fall into the other group. When we also looked at the survival outcomes in terms of both event-free survival and overall survival, we see that there is significant difference in the outcome for patients that fall into one of these groups. Most clearly for patients that fall into sphingomyelin high group, doing worse compared to the patient that fall into sphingomyelin low group. And this is our internal cohort. So with the gene expression data available, I wanted to evaluate if we could identify some signatures that are based on transcriptomic state. So we did gene expression analysis, and obviously we find different differentially expressed genes that are representative of each of these clusters. And most intriguingly, we found that the clinically poor group, which is a sphingomyelin high group, have a high enrichment of leukemic stem cell signature. And when you think of the stem cell signature, many of there are some existing resources that have looked at this differentiation trajectory from a single cell perspective. So now we wanted to apply these deconvolution methods in estimating what would be the abundances of many of these different cellular states along these trajectories based on the bulk transcriptomic data that we had. So when we do that, this tumor deconvolution analysis using CyberSort, we found that the clinically poor state with sphingomyelin high group clearly have higher abundances of different stem cell progenitor cell state that are clearly that are described in the literature. So we have leukemic stem progenitor cell state in different categories that are defined. We also have a CD34, which is described as the cellular state that are capable of making or able to genograft. In contrast, for the sphingomyelin low group, which is the clinically better group, we found they are abundant in more differentiated cell types, which we have quantified here in the bottom. And some of these signatures, they came from single cell sequencing or flow cytometry based sequencing that are done in these two publications. So in the schematic, we found that the sphingomyelin high group often have high stimness signatures, so they also don't respond to the standard of care or have higher response, high failure. And the sphingomyelin group also often has a higher abundance of differentiated cell types, and that could be what is driving these differences in terms of their response to standard of care. So now we wondered if we could use this unique gene signature in training some classifier to be able to predict the sphingolipidomic profile on some public data set. So we used a very simple support vector machine classifier by splitting the data into training and test. And when we settled on a optimized model and applied which did a fairly good job of being able to separate these two groups based on gene expression alone. When we applied this classifier on the TCGA and Beat AML cohort, which have a higher cohort of patient, we wanted to examine whether the patient that fall into one group versus the other group have differences in the outcome, as we see in the internal cohort. And this is interesting because consistent with our internal cohort, when we predicted Beat AML as well as TCGA AML, we found that patient in sphingomyelin high group do very poorly compared to patient in sphingomyelin low group. And most interestingly, this distinct outcomes by adding this biochemical information on top of what would be clinically considered, we see that the further stratification is done or is meaningful in the context of intermediate risk. So from a AML perspective, when AML patient comes to the clinic, they're often profiled for cytogenetic mutation or chromosomal abnormalities, and based on those information, there is some standard classification of risk. So there is three defined classification: favorable, adverse, or intermediate. And from a clinical perspective, intermediate risk group is often called the basket category, where they're highly heterogeneous and they do not fall into some of the good or bad risk. And what we found was, compared to the favorable and adverse risk group, our biochemical stratification of adding these lipid quantification information on top of the genomic classification, we can further stratify this intermediate ambiguous group that is very difficult to treat. So this has some clinical utility in terms of how this information could be clinically translated. And we are in the process of identifying or pruning some signatures to a bare minimum that could be easy to apply and could be done in shorter time. So summary for this, we found that a sphingolipid-focused measurement identified two distinct subgroups in AML. High sphingomyelin subtype is a previously unrecognized high-risk group with poor clinical outcome. And interestingly, when we did multivariate Cox regression analysis, we found that a sphingolipid is an independent predictor of outcome, as we see as a ratio here. So this could lead to new patient stratification in terms of how this information is used in the clinical setting. And we are currently looking at the differences of these two sphingolipid groups in terms of drug sensitivity, which could be used for providing some therapeutic options for this high-risk AML subtypes. With this, I would like to thank people. So I work closely with Kevin James and Tom Loughran. It's part of the UVA Cancer Center PO1 AML team, and funding sources that have been provided to continue this research, and some collaboration that I have been able to establish. With that, I'll take any questions that you have.
Okay, the floor is open for questions. Feel free to unmute and ask away. Fred, go ahead.
Yeah. Hey, pretty cool talk. A lot of information there. Very impressive work. So I have a question that maybe applies to both of them. Does anybody know how much variation you see in these pathways if you just looked at healthy tissue?
Oh, from a normal compared to the others?
Or just within a person, because
Yeah.
how much heterogeneity is wandering around out there that's maybe good, maybe bad, maybe harmless?
No, that is interesting. So from the lipid quantification perspective, when we looked at those sphingolipid abundances in normal versus the AML, they are separate.
Mhm.
There is not much heterogeneity in the normal context, but also our sample was limited. We had only 6 samples, normal samples. So I guess if we add more, then maybe we'll see more heterogeneity. But in the 6 that we profiled, they were not as heterogeneous as the AML cases. In the breast work that I presented, I think those transport regulators, I haven't looked carefully, but I would assume them to be heterogeneous. But I think the normal tissues or normal breast would have a distinct transport profile compared to the breast cancer case. I haven't done that analysis, but that's a good suggestion.
That's good. That makes sense. Yeah, I've been thinking about aging stuff lately, and whether or not you see increases in those sorts of heterogeneity with aging in whatever relevant tissue you can get samples from.
Oh yeah, no, absolutely. So in this AML work, we have a collaborator that looks at the aged AML, so patient older than 60 years of age, and we see a very distinct profile in those patient cohort compared to the younger cohort. So age definitely has some contribution in how biochemical pathways are rewired, or biochemical species are maintained. Yeah, no, absolutely.
Well, thanks.
Other questions?
Jim, I've got a question. This is Tom.
Go ahead.
Not so familiar with the pathway, but fumonisins, for example, could they be potential inducers of variability, of heterogeneity in the system? Fungal exposure.
We haven't looked into it. I assume any sort of extra, cellular perturbations or exposure would have differences. But that I think some of those could be examined if those clinical, like for example, GTEC has this normal RNA sequencing data available for any like thousands of samples. If there is a clinical annotation of some of those normal samples and what exposure they got and so forth, I think that would be interesting to see whether gene expression state could be predictive. Yeah.
I have a bit of a philosophical question.
Sure.
Which is in the first half of your talk, you showed us some beautiful mechanistic models that helped us understand the role of nuclear transport here. And the second half was more classic bioinformatics, stratification, pattern matching.
Right.
In your work, having worked in both of these areas, and this is a topic that comes up again and again in these meetings, how do you see the relationship between these machine learning and data-driven approaches and more mechanistic? Is there a way to put them together where they could leverage each other, or do you see these as fundamentally separate approaches that don't really communicate?
I think they need to be integrated much better than we have been able to do at the moment, because both of them add values and they have limitation on their own, right? So some of the mechanistic modeling, if we are able to integrate some of the bioinformatics derived signatures or machine learning derived outcomes, I think could be useful in identifying some mechanistic signatures that are unique. I don't think we have been able to do that, and there seem to be, I don't get it why, but seem to be some sort of friction between mechanistic modeler and bioinformatics scientist. I think if we are able to kind of bring them together, this would be advancing the field much beyond what we have been able to do so far.
I thought, Fred, you might have some ideas about looking at these two projects, how one might begin to try to integrate these approaches. I don't know for the sphingolipid pathways, which are Both the nuclear protein, the nuclear importers and the sphingolipids are not familiar pathways to me, so I'm not the person. But in the sphingolipid example, are there mechanistic models that could inform the kinds of approaches that you're taking here, or is this something that really is pure where
Yeah.
data driven is more or less the only option you've got?
No. I think some sphingolipid species have different abundances in some localized, some organelles, mostly nuclear and cytoplasm. I'm just considering very two different location, but this could be extended to other localization patterns within a cell. Some sphingolipid species have differential abundances in where they are localized. So I think there is a value in adding if we know more of those kinetic parameters into these mechanistic models and examining those localization patterns and seeing those patterns and maybe the signatures that are unique to some patient versus the others. So I think we are limited by some of the parameter where this maybe could be useful in kind of bringing in more statistical approaches in extrapolating those parameters. And Carlos Lopez in Vanderbilt and now at Altos would do some of those work. I think there is absolutely value, but I think we're limited by the kinetic parameters that make these models. And we haven't really settled on what would be the starting point, I guess.
If I had a sabbatical starting tomorrow, that's what I would work on. But I don't know how to do it. But I'm looking forward to seeing what young people are going to figure out so we can use data way more intelligently with math models to underpin them.
Thank you very much for this nice talk, Bishal. And I have this technique to how to analyze the bioinformatic things, which is the transcriptograms. And I'd like to apply to your data. So if they are public, I'll just try to do that. And then we can talk.
Yeah, we deposited that data in the GEO database. So you should have access to that, 100 of patient samples that we had profiled and cell lines.
Okay, yeah. Okay, then we can talk because when we use transcriptograms, we just reduce the noise that is usually in any measurements of many data, and that helps the signal to noise ratio, and so it improves the quality of the information that you can get. But then let me see how it is.
Yeah, one of the active objective that we have is, can we prune down those gene signatures to a bare minimum that we could functionally test and see if they have any predictive value. So I'll send you the links for the GEO database that we deposited the transcriptomic data.
I was just going to say one thing about Rita's method that's interesting is it aggregates signals into things that are maybe more amenable to mechanistic modeling.
Okay.
So you get sort of gross signals of things like promotes cell proliferation, promotes apoptosis. And so if you want to have a sort of an outcome pathway type model, which connects signals to outcomes, the way her approach aggregates maybe makes it easier to build mechanistic models of that kind. So that might be of interest.
Oh, absolutely. Absolutely. Thank you.
I'll do the first analysis, Bishal. If you send me the link, because it's easier, then it's faster, then I can make a very fast analysis, just to know if it's going to work or not. And then we can talk.
Okay. Absolutely.
Okay. Thank you. Thank you, James.
Okay, John, over to you.
In your presentation, I keep hearing you say, and then we noticed, and then we saw this, is there anything about your group of people that you would say makes you any different, maybe the way you approach this whole problem of using the model data to come up with ideas and test them? Kind of like if a couple of new people joined your group, are there things that you try to mentor them for that that allows you to do that. You as the humans, it's not the machine learning.
Yeah.
You, the humans, are learning somehow when you look at this stuff. So is there some kind of brief thing that you would like to say about your group that makes you good at that?
I trained as a cancer systems biologist, so I actually have experiences doing both experiments and data analysis, modeling, and bioinformatics. So I see a great value in kind of training people to be able to talk to both of these sides. And not many people do that. People either classify themselves as experimentalist or computational scientist, and we have not really been able to train many systems biologists. And from my experience of getting trained in that environment in both graduate school and during my postdoc, I think I would, as a mentor, be very willing to take people in, and if they have interest, be willing to train them to think beyond what they're comfortable in, because we need to get out of our comfort zone to be able to bridge that gap that we currently have between the two groups that separate the basic science, I guess.
Yeah, that's an interesting and useful response to me, because being neither a biologist nor a mathematician, computation person, I look at this stuff and say, is there something about the modeling world where if you don't have the experimental experience and background, for people that come in from the math side or the modeling side and take up biology as a subject, that you just can't recognize things in the computer models without some background and experience getting your hands wet, nasty in the laboratory.
Oh, absolutely. I have a few anecdotes. So I had a colleague who thought that they could model this system that we're working on, but their parameters required the cells to divide every 2 hours. Biologically, for eukaryotic cells, that is not possible, right? So from a mathematical perspective, you could have cells do whatever they want, but they have to be reasonable and realistic to the biological limits that are constrained in. I think if we don't have these interactions between the biological scientist and the modeler, or people trained in both space, we fall into this trap of calling the mathematical models unrealistic or unreasonable. Or then from an experimental perspective, they think that the computational people are not reachable or easy to communicate with. So I think there is a value in kind of creating this space where people feel comfortable and having this conversation that is that brings both people in.
It would, not to take 1 minute away from the marvelous work you're doing, but it might be very helpful to the community if some of you would write that story to begin to publish some stuff inside this community that talks about what you just said. It's like, we can't give up the laboratories, and maybe the real power in the modeling is what it can do for the people who came from the laboratory.
Okay, well, I want to thank you again. We had a great conversation, and it was great to see two very diverse perspectives here. Also, the connection between modeling and clinical outcome, which is not something we always see. Survival plots, diagrams, and mechanistic models are not that often in the same talk, and so seeing something which goes from the very basic science model all the way to the clinical outcomes, I think was really valuable. So I want to thank you again for coming to speak to us, and we'll look forward to come again and give us another talk on some of your other work.
Absolutely.
And I want to thank everybody who came today. Good to see you in a few weeks. Bye.
All right. Have a good day.